Hello, iGEMers. Welcome to the main stage on day 3 at the 2023 iGEM Grand Jamboree. Here's something I didn't tell you yesterday. I'm Dorothy, the Vice President of Global Development at iGEM. And my job is making friends on behalf of iGEM with people, with organizations, with different players that are related to this field. And collaboratively, we hope to build something that could be meaningful for the SynBio ecosystem. So I'm very honored to be here to introduce our long-term iGEM friends. I myself has been through a set of different iGEM roles, starting from a team PI to a judge, committee member, ambassador, and now a staff. The next keynote speaker we are having collects even more iGEM hats than me. And I believe his iGEM experience equipped him with the best understanding about what kind of technical hurdles iGEM teams are facing, struggling with, and what kind of tools could have been provided to make iGEMers' life easier. Let's welcome Alec Nielsen, co-founder and CEO of Asimov, onto the main stage.
Hello, iGEM. How's it going? Yeah, louder. How's it going? It's wonderful to see you all here. It's an honor to be back on the stage. My name's Alec. I'm a co-founder and CEO of Asimov. I did iGEM back in 2008, and probably about 15 years ago to the day, I was on the stage at the Jamboree about to present for our team. And I think I'm even more nervous today than I was then, so that doesn't go away. But today, I'm going to talk about some of our efforts at Asimov to build better tools for synthetic biology that we democratize to the community. And along the way, I'll tell you a bit of a personal story on how I found synthetic biology, or if it's not too corny, how synthetic biology found me. Rewind to the year 2006. I was an undergrad at the University of Washington in Seattle. And synthetic biology found me in a state of what the French call ennui, existential boredom, dissatisfaction. I had switched my major like a half dozen times from psychology to physics to business, electrical engineering, and more, and nothing was grabbing me. And I began to despair at the concept that I might be devoting my life to something that I wasn't passionate about. But then, as luck would have it, on a drive back from a camping trip, I stopped into a gas station, and there was a magazine on the shelf that caught my eye. It was an article of Scientific American, and the cover said, "Engineering Life. Genetic circuits will revolutionize biotech." Genetic circuits? Molecular computers? It was the coolest thing I had heard in my life. And it was written by many of the luminaries in the field, some of which are here today. Drew Endy, Joe Jacobson, George Church, Christina Smolke. And in the pages within, I learned about this burgeoning field of synthetic biology, the endeavors to program life, to build living systems that could form spatial patterns. I learned about abstractions, not thinking about things solely in terms of DNA sequences, but parts and devices and systems. And probably most impactfully for me, the article ended with this paragraph that I still feel like resonates through my bones, this first sentence that I've highlighted. The students participating in iGEM, this new student competition, may represent the first generation of biologist engineers trained to think of themselves as both. And I thought, like, this is it. This is like the thing that I've been waiting for. And it was so impactful for me that I actually still have that same issue. It's seen better days, it's got coffee stains, and it's torn, and it's held together with Scotch tape. And in case you're wondering, yes, I carry it in my pants at all times. It's for good luck. So I started to ask around at the school, iGEM, do we have a team? Sadly, we did not. But over the next year or two, we were able to pull one together. And in 2008, UW had our first iGEM team. I made many lifelong friends. It was led by the unflappable Ingrid Swanson, who was a grad student at the time. And our project was perhaps a little bit contrived, but it was to control horizontal gene transfer, shuttling plasmids from bacteria into yeast across kingdoms of life, which was something that was known to happen naturally through conjugation, we aimed to put it under the control of genetic circuits. So we had devised a circuit architecture, we had done some modeling, and did it work? Well, most of our gels looked like this. Most of our transformations failed, but that's okay. It was good practice for it to happen another 10 million times in grad school. We did recapitulate this result that we can conjugate from bacteria into yeast in a constitutive manner. And so here's the money figure, where on the right we had co-cultured E. coli with yeast, and only with co-cultures that harbored a yeast plasmid and the conjugative machinery did we get yeast survival. So we called that a win. We also had a software side project called CTOB, which isn't a super catchy name, but it was schematic to BioBrick. It was a graphical user interface for designing genetic systems using BioBricks. And you may see some echoes of this type of concept in what I'll talk about we're building at Asimov. And so here's us at the Jamboree in 2008. It was 1,200 students at the time. It was at MIT. That's us encircled. And we did not win any of the big prizes. We did not take away a whole lot of glory. Slovenia won the grand prize. Shout out to Slovenia. Are you Slovenian? Okay. I'm still, getting over it. But it was life-changing. Like, this was my first foray in the lab. From there, I went to do undergraduate synthetic biology research at the University of Washington. I started to learn more about the field. And so the next big step for me was, as I was learning about the field, I came across the work of a young professor by the name of Chris Voigt, who was bringing such a quantitative rigor to the field. Like, he was like an engineer's engineer, and I became enamored with it. And I targeted Chris specifically for grad school and went to work with him for my PhD. And over the course of 6 years, we collaborated with a professor by the name of Doug Densmore at Boston University to build a number of things. I worked on machine learning applications for biosecurity, reprogramming CRISPR to make a programmable transcription factor. But the main thrust of what I worked on and collaborated on as a grad student was this genetic circuit design platform called Cello. So since I'm at iGEM, I got to kind of like do some nerd out on genetic circuits. Cello was the culmination of many years that we had spent in the lab developing very extensively engineered, well-characterized, insulated genetic logic gates. So modular, in fact, that we could change what genetic context they were in and actually predict how they would function. That was very important. And we integrated that with computational tools to simulate genetic circuits, to automate their design, so that we could push the envelope and design genetic circuits that would be far beyond what a human could do by hand. And we built hundreds of circuits over the years using Cello, and it had a pretty good success rate. 75% of what the computer compiled and output worked on a first pass. And my favorite example that I'll walk you through, so this is like 6 years of my life in one slide. We built a digital display. I love this because it's a really visual application. And so here's the concept. Like, on a digital clock or a calculator, you've got these digital displays, right? And what those do is they convert a binary electrical input into a visual decimal digit output. And you can buy these off the shelf at Radio Shack if you're an electronic engineer. And we wanted to build a version of this inside cells. So what Cello allowed us to do was to specify this input-output behavior using a high-level behavioral language. And then Cello uses a number of electronic design algorithms to map that into a logic diagram and goes through a series of transformations that ultimately ends up with a circuit that only uses NOR gates. NOR gates are a type of logic gate that are very special. They're Boolean complete. Any digital circuit you can conceive of, you can build with just NOR gates. And importantly, they're very genetically tractable in bacteria. It's basically two tandem repressible promoters driving a repressor, and then you can connect these things up. And so at this point, Cello has only generated a mathematical abstraction. There's nothing genetic about this. So this is where our genetic toolkit came in. We had developed this orthogonal library of repressors that were insulated that we could mix and match. And we can start to assign those genetic parts to this circuit diagram. So now I've colored this diagram based on what gene has been assigned to it. And as you can probably infer, there's like a very large number of ways you could color this. There's a very large number of parts assignments, and they're not all created equal because of like the quantitative behavior of the transfer functions. And so this is a combinatorial optimization problem where we, in silico, screen lots of parts assignments and simulate all of them. And so what we're doing here is modeling the internal transcriptional state of every node in this circuit. And throughout this process, we can then output the best predicted parts assignment for this genetic circuit, shown here in schematic form. So if you squint, you can see promoters and repressor CDSs, et cetera. And at this point, it is one-to-one mappable to a DNA sequence. So like in some sense, Cello is a compiler that starts with a high-level behavioral spec and then gives you a DNA sequence output. You have to now build these things when we're talking about like 10-gene plasmids, so it's non-trivial. But once we do that, we can load them into E. coli. And just like in the electronic version, that's actually like a fluidic display. It's 7 different segments that have a liquid crystal in it, and when you apply a voltage, it will crystallize the fluid and make it opaque. And so we did something similar. We built this fluidic display that we could load our 7 strains of genetically engineered E. coli into, and then rather than apply a binary electrical input, we can apply a binary chemical input in the form of common chemical inducers. And what's happening is the cells sense those molecules, decode that information, and ultimately output the presence or absence of green fluorescent protein, and we get the digits 0 through 9. This is not an industrially useful application by any stretch, but I love it because it is very visual, and it communicates what is possible when you bring together well-engineered genetic components with computational models and tools. And this is still one of the most complex genetic systems that's ever been built, something like 70,000 base pairs, 62 genes, not just overexpressing genes, but coordinating them in a symphony where they can switch each other on and off. And so I was on the academic path. I was finishing up my PhD and was planning to go do the academia thing, and then I reflected on the past decade, since I had first learned about genetic circuits to where I was then. And I show this as a path. It almost felt like following a trail of clues, where the clues were like pointing to the next most interesting thing to work on, with the most interesting people. And over the course of many sleepless nights, ultimately became convinced that starting a company was the next most interesting thing to work on with the most interesting people. So teamed up with my thesis advisor, Chris Voigt, Doug Densmore, and a classmate from MIT named Raja. And we started Asimov in 2017. We've raised venture funding, support from DARPA. And if you fast forward to today, we're just over 90 people in Boston. And it's a really eclectic mix of people from all different backgrounds. I often will cornily describe it as a culture of recombination because we are recombining all of these backgrounds and skill sets. Synthetic biology, machine learning, UI, UX, and of all the things that we've built at Asimov, the team is what I'm personally most proud of. And so, what are we building and why are we building it? Well, I think like many in this room, we believe that engineered biology has the potential to increase the quality of life for a very large number of humans. And so our mission is to enable those biotechnologies. And working backwards from that, what's the best way to do it? Well, we concluded that genetic designers deserve better design tools. Not just need better design tools, but actually deserve, right? We're working on really hard problems, but that will impact health, nutrition, the planet, climate, and it's really hard. You all know that. And I had seen Drew Endy give this talk in 2009 with this analogy that just burned itself in my brain, which is if you look at other engineering fields, over the decades, they mature, and they move from manual design to computer-aided design. I love this image of Intel in 1970, where engineers are designing a circuit by hand, carving out a diagram on a vinyl sheet that they'll then use as a stencil to pattern silicon using UV light. And these days, you carry around 10 billion transistors on you at all times. That's, you need better tools than a scalpel for that. And so on the right is a state-of-the-art computer-aided design platform for electronic circuits. If you look at genetic engineering, I'd say we're still kind of in the dark ages. It's still more trial and error than engineering. The only ubiquitous software tool that you see used across genetic engineering labs is a DNA sequence viewer, which is a useful tool, don't get me wrong, but it's more akin to a text editor than what you'd expect with computer-aided design tools, right? You can't simulate, you can't optimize, you can't analyze these systems that you're building. So that's what we're building at Asimov, is a computer-aided design platform for genetic designers. But there's a really interesting kind of twist to all of this, which is we build software, of course. But importantly, the software is integrated at a deep level with all of this biology that we develop at the company. This includes chassis. This includes genetic parts that we characterize in those chassis, models. And the business model is a little bit different than like the classic synthetic biology business model in that we're not a CRO, we're not a contract research organization that does services, and we also don't make our own end products. Instead, this suite of cells, genes, and software is something that we provide to other companies and labs to level up their ability to do genetic engineering. And right now, our focus is exclusively pharma applications, which means overwhelmingly mammalian chassis like human cells, Chinese hamster ovary cells. But the applications span protein biologics like antibodies, cell and gene therapy, RNA, and we've garnered 30 commercial partnerships. In the future, we're interested in expanding into other non-pharma domains as well. I'll walk through some of the tech stack really quick. So genetic parts, which you are all, I'm sure, intimately familiar with the concept. We develop thousands of genetic parts that we characterize in different chassis to control different aspects of cell function. And so we've developed this off-the-shelf toolkit for programming mammalian cells. You could kind of think of it as controlling the central dogma and its general purpose. We're also starting to develop more targeted genetic parts for specific applications. Here's one of my favorite recent examples, where we developed a large DNA language model that generates DNA sequences that are tissue-specific promoters. So here's the concept. Gene therapy, which is this amazing, very sci-fi-seeming new therapeutic modality where you're essentially patching the human genome, delivering a functional copy of a gene to treat some inherited genetic disorder or disease, is currently being met with all sorts of challenges in the clinic. A lot of toxicity, even patient deaths, and it's holding things back. Why are things toxic? Well, to give you a sense of the problem, if you're trying to treat muscular dystrophy, you want to deliver and express the gene dystrophin in skeletal muscle, but not in other tissues like the liver. That's a gene that has no business being expressed in the liver and can result in toxicity. So one way to mitigate the toxicity of these gene therapies is to have precise control over what tissues these payloads are expressed in. And so this DNA language model allows us to specify that we want something on in, for example, a heart, and off in every other tissue, including the liver, and then it outputs a DNA sequence, which we then validate in animals. And a picture tells 1,000 words. The lower right are 3 mice that have been dosed with this AAV gene therapy with a tissue-specific promoter, and the hearts are literally glowing with no detectable expression anywhere else. And I want to stress, this was actually the first round design. We didn't go through multiple cycles of design, build, test, and so we're really excited about where to take this next. We build a lot of models at Asimov across the entire scale of biology, from predicting gene expression to phenotypes to metabolic maps of the cell to even bioreactors, where many of our systems are deployed. And then lastly is our software interface, which we call kernel. And I want to take an extra second to talk about what kernel is. Kernel is the software that we're building for teams to collaborate on genetic engineering projects. And so it includes some of the things that you might expect. It includes, for example, the ability to create a team and invite other members into that team. That could be a company or an academic lab. We've got an electronic lab notebook where you can create notebook files, protocols. You can organize things into projects and folders, things that you would expect. We've also built a genetic design interface that also includes things that you're probably familiar with, like a DNA sequence viewer. But we aim to move beyond pure DNA sequence viewing. And one of the things that we've put front and center is this design schematic, where you can drag and drop parts. We've integrated with hundreds of thousands of genetic parts across databases, including the iGEM registry. Big shout out to the iGEM team who helped us do that. And I can tell you, as someone who has designed literally thousands of plasmids, it is a very fun, fast, flexible way to design. And in fact, it matches much more closely to how we communicate and collaborate as synthetic biologists. And so you can code and optimize, you can tailor the visuals for a presentation. But the last thing that I'll show you is what I would consider the advanced capabilities. So given that genetic design up top, we've integrated what we call the genetic simulator. This is a data-driven model that predicts the cellular, the biophysical phenomena that arise from that DNA sequence up top. All right, so you can mix and match and redesign that schematic and then simulate, and you'll get a new simulation that models things like transcription, translation, concentrations of RNA and protein, regulation, molecular complexation. It's tailored at the moment for mammalian systems, where we've collected the most data, but we're interested and excited to expand that into other organisms. And I will say it's far from perfect, right? But I will note that it is systematically improvable with data, and it is far better than what we've had to use historically, which is our own even more imperfect mental models. And so this is a direction that we're excited about, new abstractions, getting back to compilation, so not just manual design and then simulating what it does to sanity check it, but new tools that can allow a user to specify what they want and get a DNA sequence out. That's on the horizon. And then lastly, we launched this week. It's our AI assistant and knowledge base. And so the concept here is an AI model that has access to all of your organization's lab notebooks and protocols and genetic constructs, papers that you've uploaded, curated knowledge that we've put into it as well, like our genetic design rules. And you can ask it questions like, what primers did I use to amplify Cas9 last week? Who was the most recent person to do a transfection? What was the best part out of this genome part mining paper? And it'll give you results. And we really like this idea that you could have an AI co-pilot that helps with every single stage of genetic engineering R&D, from conception to plasmid design and debugging, to understanding experimental data and suggesting new experiments to do, to understanding the literature. And of course, we've got many passionate biosecurity advocates at Asimov, and so we're thinking about strategies to get the benefits of democratized synthetic biology knowledge without the risks. And we've started to talk to certain teams about collaborations in that vein. So if you're interested, please reach out. If you find this interesting, I want to give a quick plug for the blog. We've been writing essays about this technology that we're building. We're trying to make it accessible and technically grounded. Check it out. And I also want to give a plug for something that will be launching in the next couple of months, Asimov Press. This is an editorially independent media organization that's going to be writing books and publishing essays. It's created and helmed by Nico McCarty, founding editor. Why are we doing this? Because we want more inflows into the bioeconomy. Inflows of what? People, capital, ideas. The field needs more new ideas, attention. So if you want to sign up, there's a QR code there, and stay tuned. And then lastly, I want to finish with what we're thinking for our 2024 partnership with iGEM. So I'm proud to say that I've gone over the years from an excited outsider to an iGEM team member, to an advisor, to a judge, to a track organizer, to now a sponsor. iGEM changed my life. I really say that without any bluster. And we want to give back. We want to help grow this community. And so in 2024, as we did in 2023, we're going to be manufacturing the distribution, all of the DNA kit plates and the measurement kit, and shipping those to teams across the world. We're going to be providing more synthetic biology support. We're going to be open sourcing more genetic parts and design rules, giving access to some of our expert synthetic biologists to help with teams. And then secondly, in 2023, we had a small group of iGEM beta testers for kernel. Thank you for all the feedback that we received. In 2024, we're going to go way bigger. Any iGEM team, whether you're working on mammalian or not, if you're interested in these tools, let us know, and we'll get you access. Same goes for academic labs, and we're going to be launching an initiative called Kernel in the Classroom for educators. QR code if you want to sign up and learn more. So with that, I want to thank you. This is, again, it has been an honor. I feel like I've come full circle 15 years later. And if I could just really quick, because I want to remember this forever, if I could do a quick panoramic shot. Everyone say cheese or fromage or your own language. Let's go. Beautiful. I will cherish this forever as well. Thank you, iGEM.
Thank you, Alec.